There we go. I think we should be going. Does it look like it's recording from your end?
Yes, it
Okay,
definitely is.
great. Okay, I just wanted to check because I'm
Yeah.
sharing my screen and it's not giving me my normal pop-up. Um, great. So thanks all for coming today. We're going to talk a little bit about mismatch repair deficiency variants, or like insertions and deletions of variable length within MMR genes. Want to cover how are we currently reporting them, how do we feel we should be reporting them, and if we can, as a community, kind of do a better job with this kind of very small subset of variants, which I find really interesting because it's almost an illustration of itself. Illustration of mismatch repair deficiency, in that you have these regions of instability where you can see insertions or deletions of variable length. So we'll go over an example of that today, and really hoping for a good, vibrant discussion about our current practices and how to move forward, and kind of help guide our database and communities and things like that too. So really looking forward to this one. So I know I'm probably speaking to the choir here, however, or preaching to the choir, but mismatch repair genes, the ones that we typically assay, are MSH2, MSH6, MLH1, and PMS2, and they work together. So MSH2 and MSH3 and 6 work together and as their mut S homologs during mismatch repair. And then mut L homologs, MLH1 and PMS2, kind of work together and dimerize. So I'm not going to go too much into the mechanism here, but know that these are really important genes for repairing your DNA, which mutates all the time, especially in cancers. And so when you have a deficiency or a loss of function variant that is driving cancer, you tend to have more mutations. So you'll have a higher tumor mutational burden, but also we'll find deficiency in these genes through IHC and through other microsatellite instability type assays. So when these variants happen in the germline, pathogenic variants can cause hereditary cancer syndromes like Lynch syndrome, and when they're somatic, they enhance genomic instability, like we were talking about. And this genomic instability may confer sensitivity to immunotherapy or checkpoint inhibitors. So here's a case example I've de-identified and kind of simplified for this discussion. This is an adult patient with colon adenocarcinoma with a known MSH2 truncating variant that was called pathogenic at 30% VAF. The tumor was 60% tumor purity by pathology, and this was classified as microsatellite unstable, or MSI high, by ADILA testing outside of our NGS test. And so we knew this was MSI high coming in, but we had this set of variants here all within the same locus in MSH6. So we can see duplications of anywhere between this C dot here, so C dot 3258 through 3261 at different lengths. So we saw this, I immediately wanted to look at the pileup for our data. And when I looked at the pileup, I realized that we had a lot more variants than just these couple that were listed here. they were just sitting underneath our assay's limit of detection. And so you can see that 2 of these variants are at only around 3% VAF, so they're right at the boundary of our limit of detection. But when I look at the pileup, you can actually see quite a few more variants, including an insertion of 6 cytosines, all the way. So you can see 1 here, that's 6 cytosines. You can see 5, 4, 3, 2, and then if you go down, I think there were 1 or 2 1s there too, or 1 or 2 like insertions. Then you can also see a couple deletions right at the same locus. So this is an interesting phenomenon, and we've seen it on a number of instances. So all these variants are at the same CDOT location. The IGV pileup made it clear that the inserted sequences are all cytosines, and we saw the insertion anywhere from 1 to 6 cytosines, as well as the deletion of 1 to 2 cytosines within that region. You can see that in the reference sequence in the bottom here, that there are in the reference sequence 7 cytosines in a row. And all the variants that were not reported were present, but they were under our limit of detection. So this kind of causes some confusion as to, okay, well, now that we understand what's going on, what should be reported? So what do we do with this variable length insertion or deletion? Do we classify each independent variant of every length and report them out separately or together after that? If we do, then the different lengths of insertions will have different classifications likely. If something is in frame and not truncating, that may have a very different classification independently than something that is a truncating variant in a tumor suppressor gene and an MMR gene. So just a couple of things to consider here. And then also when we're using our oncogenicity standards, we have an in-frame duplication. This is not present in cancer hotspots at all. It's not, it doesn't have a Revel score, but it is absent in population databases, which according to these standards would put it out of the US. Whereas a frame shift is a null variant in a tumor suppressor, which gives it some very strong evidence moving toward oncogenic, as well as being absent in population databases. And so we kind of see that the different variants alone have different classifications. So any comments, discussion at this point? If not, we can move along. So I did put together a little survey that I can send out. So I'll launch that right now for our team on this call to answer. And then when we have those answers, I'll go ahead and share those with the team and we can discuss a little bit more.
Hey, Beth, I have a question is it okay while people are answering the survey?
Of course. Yeah, no, this is perfect.
I'll put my video on. When you're interpreting this, so I guess my question is how much confidence do we have that all of those are actually present and that it's not, like if you have a run of cytosines, that it's possible that you're seeing all these different variants with all these different size insertions or deletions, but is it also possible that there's polymerase skipping in the NGS that you've got? I think that contributes to whether you call them together or apart and how you call them.
Yeah, no, I think that's a really good point, and that's probably why, you know, in our final report, we only pointed out the ones that cross that BAF threshold. So even though we could see them in the pileup, and we were pretty confident they're there if we have, you know, 5 or 6 replicates of that size. But I think that we ultimately just said this is what we are validated to report, and we report it out as such. Like we see these variants in this region, and together they are classified this way. So that's how we did it. However, in our system is not set up to combine variants in that way. So it was a very manual process. So it's not perfect. In our system, we have to classify each variant independently in order to move the case forward. So we ended up with our in-frame variants being classified as the VUS, and then our truncating variants being classified as at least likely pathogenic or likely oncogenic moving forward.
Right. Which probably for patient management doesn't matter, but it's interesting.
Yeah, it feels like the process there's potential for better, more clear streamlining of information.
Yeah, I mean it's interesting because it's there's clearly some kind of slipping happening, whether it's the polymerase in the the NGS itself, or whether it's happening in the cell, and then we're detecting all these different slips. And so I think for a scientist, it's interesting to know what's happening on a molecular level. For the clinician, it's a different question of what's the most important thing for them to know and understand, like how much detail they need or want. I don't know.
Or yeah, that's totally legitimate, and that's part of why I said additional considerations for clinicians and patients. I mean, really, all they're looking for is this MMR deficient?
Right.
Yes or no, right?
Yep.
Well, most clinicians, I think, but
yeah, I think there's some that certainly would want more information and want to understand exactly what's going on, but maybe the minority of clinicians, I don't know.
Yeah. Yep. Sounds good. Manuela, I see your hand is up.
Yeah, I was wondering, I mean, it goes into the same direction. It's the question of slippage. Do you actually filter for slippage effect in your protocols, in the bioinformatics analysis?
I know that we have like UMIs and UDIs in every assay, so it's a little bit more precise, and we're not going to be duplicating a slippage, like a read with the slippage necessarily. So I think that controls it to some extent. And other than that, I'm not really sure.
So yeah, we tried to look at it at a certain point and actually decided against the slippage filtering with respect to what we report. That was a research setting though. But I think it's quite important to see like what is clinically relevant. And I think here the most relevant variant, if it's above a certain allele frequency, is I guess the most important one, because that's where most of the time decisions are taken based on. So it's I don't know. I don't know any discussion which was based on a general classification. It's usually really the variant itself. So yeah, I would still go for single variants presenting in the clinical case, but yeah, additional information if wanted.
Sounds good. All right.
Can I ask a question? Hi.
Of course.
This is Jalin. I'm the molecular pathologist. For this, I wonder what kind of technique or sequencing platform it's used for this sequencing. It's an iron torin or it's a Lumina?
This is I believe this is like a custom made IDT panel that we run on Illumina.
Illumina, I see. And it's the correlation with the mismatch repair protein immunostain or TMB or MSI.
Yeah, we do MSI testing with all of our colorectal NGS specimens. If we have, I mean, really our MSI testing, it requires really minimal DNA, so we can run that on pretty much anything. And then I think it's, most of these do have IHC, but not all of them have IHC at our institution because Mayo is a large reference laboratory.
I see.
Yeah.
For this, do we have a TMB or MSI data or mismatch repair protein immunostain data?
Mm-hmm. Yeah, so this one was both MSH2 and MSH6 deficient.
Oh, God.
Yep.
Thank you very much.
Mm-hmm. Great. So I'm now sharing our poll results, and looks like thank you to those who answered the questions. Looks like 3 of 4 people who answered so that they would report and interpret all the variants at this locus together, or it sounds like potentially like pick a variant that would mean the most and kind of explain it in the interpretation. But yeah, I was kind of wondering like what the current practices are around variants like this. So if anyone else has a different experience with these, I would love to hear it. If not, we can move the discussion along. But I thought that this was probably one of the more important points. All right.
Sorry, I have one more question regarding this case. I don't remember the variant allele frequency, but this was kind of borderline towards germline. Are you kind of discussing that as well as a prospective or potential Lynch syndrome then?
Our boundaries of like calling something potential Lynch syndrome are usually we're looking for something that's closer to that 50%. I mean, we could, I think it's more at like director discretion to call out something that's as low as 30%. However, it's not unheard of. But I don't think we did in this case call out a potential for Lynch.
But what was the tumor purity? That was 60, so
60, yeah.
Yeah, and then you have 30, so that could be that's kind of borderline.
Yeah,
And
it is. I could show you these ones.
Yeah.
So the MSH2 variant was right at 30% on the dot. And then the highest of these MSH6 variants was at 23%.
Okay. Presumably not, but I guess here, so maybe as a comment, the discussion in our molecular tumor board would always go towards the human geneticist, and the common question would be, do they have polyps? Do they have anything additional information on the patient, and how young is the patient? Because if it's Lynch, it would show up quite early.
Right.
And you would see additional markers like in the pathology of the colon itself.
Right. And isn't germline testing recommended for most, if not all, colon cancer patients? I want to say that that's part of a guideline somewhere, but I'm not a colon expert.
Me neither. I think we always discuss it, but yeah, that yeah. I wouldn't want to say anything on that. That would be
right. Okay.
wrong.
That sounds good. All right. And then what are thoughts about how variants like this should appear on a report? Like should we say, you know, loss of function variants at this locus on MSH6 were identified, and leave it at that? Should we talk a little bit about like this variable length insertion and deletion in the report? What are thoughts on that? Or is that too technical?
I mean, I think you can have that as a comment somewhere, but I think the sort of bottom line is that you're pretty sure there's a loss of function in this gene.
Right. Yeah.
So like whatever way, it's hard to say like which mutation do you report, or do you report a couple of them, or how do you decide to do that, and then have a note that says, like, maybe there's a couple of them reported and have a note that says, you know, from a technical standpoint, there were multiple mutations, like this is a run of cytosine, like give the technical details so that someone could look at them if they want to understand more deeply. I don't know.
Yeah. I think that's where I would prefer it to go, just because I think that, like, in order to accommodate all the different levels of expertise that get our reports, it would make the most sense to, you know, maybe the first two sentences be very concise and say loss of function in these genes were identified, and then go on to list those variants, and then go on to say, by the way, these may all be related or part of the same event.
Yeah, a single event, exactly.
Yeah. Charlene, were you coming off of mute?
I think I muted myself. Yeah, but I have no. Yeah, thank you.
Great. So, and then this is a great discussion about considerations for clinicians, patients, and how to kind of like right fit those reports. And sorry to pick on you, Cheryl, but how would somebody at N of 1 take this and say, like, this is, how would somebody at N of 1 interpret this kind of variant, or kind of think about how would they put the pieces together, or how would your group, following your protocols and things, look at this?
I mean, based on our protocols, they would be, so the decision of whether to report them together or apart, we disengage from. So because we're not doing the sequencing. So if this were submitted to, this could be submitted to us one of two ways. It could come to us as a single mutation or a handful of mutations, which we would interpret independently. And so we would have interpreted probably those frame shifts as pathogenic, and the in-frame deletion as a VUS. Or it could be, you know, upstream of us, you guys or whoever could decide this is really a single event. We're just going to report it as one frame shift mutation and that's it. And then we would interpret it that way. But we do handle each mutation individually. So if you gave us 30 of them, we would report 30 of them and classify them individually. Yeah,
that makes sense. It's easier to automate that way too.
Right, exactly.
But I'd like to think that there's a space somewhere where we'd be able to say, okay, well, these are regions of known, like, hypermutation or of known instability within,
for sure,
you know, MMR genes, and just be like, you know, maybe not to like advocate for it to be a hotspot or anything like that necessarily, but like an identified region of instability, and maybe I don't know, maybe it's something that we can talk about at the I don't know how many here will be at the unconference for CGC, but I think this could be a really fun topic to try to tackle. Like, how do we
Yeah.
discuss
I mean, I
these?
think there's these, and there's also the situation in genes like AR where there are polyglutamine repeats that are actually completely polymorphic. And so those can sometimes end up getting interpreted as pathogenic because there are losses and gains, and sometimes frame shift if it's, mostly losses and gains just because it's polymorphic. But that's, the opposite situation where you have something that actually, it's not even artifact, it's just polymorphic.
Right.
So.
And then, no, that makes sense. Then you have a variant scientist who's looking at that, and would be nice to say like, oh yes, this is polymorphic. But I think that the AR polyglutamines, those are those well documented in databases as like
there's
VTB?
A mix. Like, I think if you look in certain databases, a lot of them are classified as pathogenic.
Oh,
so
okay.
Yeah.
Good to know.
But yeah, for this type of situation, yeah, I mean, I think at the end of the day, it's different depending on whether you have someone who's scientifically inclined who really wants to know exactly what's going on versus the clinician who says, is it MMR or not? And I think that can inform how you end up doing the report. From a database standpoint, yes, in order to automate, we typically we do it mutation by mutation. However, it's also worth noting another type of complex mutation, BRCA reversions, that are additionally complex in terms of especially in liquid biopsy, we see many mutations that it's hard to know, like, where does this thing stand now? Is this driving it or not? Because there are a whole bunch of frame shifts and a bunch of missense mutations and maybe some in-frame deletions that then remove the whole thing. And are they restoring function? And therefore, is this thing going to be sensitive or not? It's very difficult to say.
It's really difficult, especially when you're doing short read sequencing. It's really difficult to look at reversion mutations. So. Yeah. So hopefully the next 5 years or so will be really exciting for that as people start to employ more long read sequencing.
Yeah. I'm thinking about two other possibilities. If I remember correctly, MSH2 has a pseudogene. Could this be caused by pseudogene?
Is that MSH2 with the pseudogene? I know like PMS2 has a pseudogene.
That's MSH2 has a pseudogene.
It has
possibility I'm thinking of it's probably just a mapping issue. If that's the case, probably if we do like a single sequencing, just amplify this spot, may get a very clean read.
That could clear up the slippage too, I would think, right?
Yeah, because it's so focused here and in the homopolymer region. Yeah, I'm thinking about maybe the single sequencing can clean up.
Yeah, and this is MSH6, but we have seen these in MSH2 as well in our lab. There's 1 or 2 areas of MSH2 that we've seen the same kind of thing repeatedly in MSI high cases. I mean, it's very rare to see this, but we've seen these variants. The MSH6 is probably the most common area where we've seen these variable length insertions and deletions, but I have seen it at MSH2 also, and that's a really good consideration is like pseudogenes, because we know that you said MSH2, PMS2, we have to be very careful with PMS2, as does everybody, because 5 of the 15 exons are part of the pseudogenes. So there's a lot of stanger that goes on with that. if we see something like this in PMS2. Or we can just revert and say, let's do IHC instead, and figure out if it's actually causing a loss of function. Oh, that's a really good consideration. Thank you. Great. Awesome. So I think that my last question was about should there be a way to store this kind of data in publicly available databases or knowledge bases? Would it be appropriate to put this, or to advocate to put something like this into a cancer hotspot with some information, or to, you know, talk to, let's say, like the civic group or the OncoKB group to say, oh, this is a region that we typically see a lot of variants and like co-occurring variants within the same locus. Or should these even be stored as, oh no, they should be stored as individual variants because of the automation thing, but should there be a flag in our databases that says, if you see this, then, you know, really check your reads and see if there are others in the string of cytosines?
Yeah, it's a really good question. I mean, it's certainly a little bit complicated with the databases. I'm sorry, I have like 100 thoughts in my head right now. One of them is that in terms of determining oncogenicity, if you have the frame shifts, then basically everything else doesn't matter because you have a tumor suppressor with a frame shift mutation that's upstream of an NMD, it's going to be null.
Right.
It's really the in-frame deletions that cause the confusion.
Mm-hmm.
But if you have them in the if you have them in the context of a bunch of frame shifts, that again almost doesn't matter clinically, but it's
right.
Yeah. And at the end of the day, are those actually, like, if those are present and they're in a small subset of clones and they're incidental, they might not be oncogenic individually.
Right. That makes sense. And that, yeah, they might not be individually oncogenic. They could be, and I think that a couple of our laboratory directors were saying, well, if we have an MSH2 mutation, does it even matter to report the MSH6, because it's not going to, it's not going to affect the patient outcome. And really, this is likely not the driver, is probably a passenger to the microsatellite instability, and kind of an illustration of that within the gene.
Yeah, I mean, there's no way to know, like the original one could have been one of those, and then you get the feedback, right? So it's actually kind of impossible to know.
Yeah, it's a chicken or the egg, right?
Yeah.
Which came first? Cool. All right. Well, really, that's all I had prepared for today, but I wanted to give anybody a chance if there are any other thoughts or comments that you want to bring up. Otherwise, we can give you all some time back to your day. All right. Hearing none, thank you. I'm going to say thanks to you all for participating in the discussion and for contributing to different considerations that we need to have when we're looking at variants like this. So thanks so much for joining today, and we'll see you all in a couple weeks.
Thank you.
Thank you. Thanks for preparing.
You bet. Bye, everyone.
Bye.